So it's my pleasure and honor today to present the 2021 B.A. Stone Award for Excellence in Plant Polysaccharide Biochemistry. And the B.A. Stone Award was conceived in honor of Professor Bruce Stone, who passed a number of years ago now, but the award was conceived to recognize outstanding contributions to our understanding of cell wall and more general carbohydrate biochemistry. So reflecting Bruce's international outlook, the Stone Award is open to scientists from countries all around the world. The awardee should have made at least one major contribution in a field that has achieved international recognition. The Stone Award includes a medal you see on the right there and an honorarium. And in keeping with tradition, the B.A. Stone Award is presented publicly at an appropriate international scientific conference. It's been at the CBM and also the plant cell wall meetings. So keeping, again, in mind Bruce's broad view and kind of the interleaving of both polysaccharide biochemistry and plants, it's really fitting to present it here at the CBM. And one more piece of information about the award. It was initiated by Megazyme Limited, actually by Dr. Barry McCleary, which continues to be sponsored by Neogen Corporation, which acquired Megazyme just this year. And I really would like to express my thanks to both Barry McCleary and Eileen Murphy for their continued support of the B.A. Stone Award. Without them, the award would not be what it is. And just a couple of words about Bruce Stone, for those of you who may not have had the pleasure of meeting Bruce. He was a professor of biochemistry at La Trobe University in Australia. He was really an international expert on serial cell wall biochemistry and polysaccharide chemistry. And one of his major life passions was 1,3-glucans and mixed linkage glucans. So many of you will know that Bruce has published a major book, a major tome in the area on the broad distribution of 1,3-glucans in nature. Bruce always had a multidisciplinary outlook and developed a lot of techniques for polysaccharide analysis and really influenced the field in many, many ways. And one of the things that was very interesting about Bruce is he did have this international outlook. He was always interested in interacting with younger people. And personally, I could say I had the pleasure of meeting Bruce in his later years, and our interactions led to really the genesis of Cassiopeia, because Bruce was really interested in developing a comprehensive encyclopedia of carbohydrate active enzymes. So without Bruce, certainly the field wouldn't be what it is today. And it's really a pleasure to be able to present this award in his honor. And I need, again, to thank Dr. Barry McCleary for this text, which I borrowed from a previous presentation. So the previous B.A. Stone awardees are this list here. I won't list all these names, but you can see there's a great number of very well-respected and influential people in our field. And I would like to just say that acknowledge everyone here on this list, because the 2021 award was decided by committee. We had a tough task in front of us because of so many potential and great candidates in our field. But through some back and forths, and of course with COVID, it was a little challenging, but we did it all over email. And I must acknowledge the work of the committee in coming up with the 2021 awardee. So really that leads to the question, who will it be in 2021? And I'll start out with a bit of a hint. I'm not very good at keeping secrets, but this person, the 2021 awardee, generated an acronym of really incredible impact to the field. And this acronym was defined by this person and really defines this person, I think, as the 2021 B.A. Stone awardee. So in terms of numbers, as I've said at the beginning, one of the criteria, or the criterion for the 2021 for the B.A. Stone award, is that the awardee should have made outstanding contributions to our understanding of cell wall and more general carbohydrate biochemistry. And just in terms of metrics, I pulled these from the Web of Science the other day, really phenomenal publication record, well over 500, approaching 600 publications, a tremendous H index. And as you could see, this person, the impact that they've had on the field has just been exponentially increasing, much like the number of sequences in GenBank. So again, by introduction, I thought I would take us back to 1989, really before this person's impact on the field was realized. And 1989 for me is a bit of a genesis event, so I'd like to just explain that in this slide. And that was the publication of a paper by this individual. They were the first author, and it was the presentation of cellulase families revealed by hydrophobic cluster analysis. And for those of you who don't know what hydrophobic cluster analysis is, it's a way to read from primary sequence. It's a way to lay out those primary sequences and read secondary structural information from those sequences in the absence of tertiary structure, of 3D structure. So this was really insightful at the time, and it led to the establishment of the first cellulase families, as it says there, cellulase families as they were known at that time, A through F. And subsequent to that, many of you might be more familiar with some of these papers, where this individual developed then the classification of glycosyl hydrolases, of course, moving beyond the degradation of polysaccharides, moving into the biosynthesis with a classification of glycosyl transferases, later on polysaccharide lyases, and even auxiliary enzymes. And of course, many of you know that this person's been is classified also carbohydrate binding modules and carbohydrate esterases. So, you know, but again, as I said, this is sort of a genesis moment for me, really, this insightful look at sequences and realizing that maybe we would be well served by grouping these enzymes by families. So the 2021 BA Stone awardee really took us beyond the concept of EC numbers, enzyme classification numbers, or enzyme commission numbers, which was really the predominant way to classify enzymes at that time. And in doing so, this person provided a framework really for a whole field and many fields to relate protein structure and function. And I think that the impact has been so widespread, we sort of lose sight of that because it's really endemic. We use words like GH family 2 without even thinking of it. And of course, this was not always the case. So with a sort of gratuitous musical reference here, I've put the top 10 hits from this individual, where they've informed plant polysaccharide biochemistry all the way from primary production to catabolism or degradation. And of course, this is one of the watershed papers that I think many of you will know, but note that that ranks only as the number 5 top hit. And really, I think what's very telling about this individual's contribution is they've been involved in really a number of major papers in the field, in different fields, where this classification has informed biochemistry and biology. First and foremost, the genome of the first tree ever sequenced, Populus trichocarpa, a very important paper for plant biologists. And of course, more recently, this individual has had a lot of insight and seen that the microbiome and many environmental bacterial niches are really ripe for understanding and classification in terms of family structure. So, yes. So again, really, this just highlights the far-reaching impact that this individual has had on both biosynthesis and biodegradation of plant polysaccharides. But before I reveal who this person is, and I think many of you might know already, I just wanted to highlight that the awardee is first and foremost a biochemist. And some of their early work that predates their work as a bioinformatician was also very impactful on the field. So the awardee worked with colloidal gold labeling, of cellulases and showed that indeed these cellulases seem to bind to the flat faces of cellulose crystals. And they were the first to provide evidence that enzymes work specifically from either the non-reducing or reducing end, non-reducing end in this case, by showing through electron microscopy that cellulases sharpen the ends of crystals. And of course, this is one thing that, again, maybe we take for granted, that we've forgotten over these many years, but again, very impactful. And of course, this individual has gone on to also continue to participate and lead many characterization studies involving glycoside hydrolases and so on. So even though you might be tempted to classify them as a bioinformatician, I would say they're really a biochemist at heart. And I think that the person always, as even evidence today, asks some very insightful and probing biochemical questions in meetings, and certainly something I've realized and appreciated over the years. So without further ado, it's my pleasure to award the 2021 B.A. Stone Award for Excellence in Plant Polysaccharide Biochemistry to Dr. Bernhard Henrissat. And of course, the acronym is KZ. It's a word that we use, or an acronym we use all the time in as itself. Kzymes, Kzymology, Kzomes, it's everywhere. And we thank Bernie for that. So my sincere congratulations, Bernie, very well deserved.
Yeah. Thank you, Harry. I think, you know, just this is just amazing. I don't know really what to say. You know, this is an award, or this is a prize, or this is a medal that means a lot. It means a lot because of the name, you know, just the Bruce Stone. Bruce wanted to do a postdoc with me 20 years ago. And that was one of his last goals, was to come to Marseille and do a postdoc with me. You know, so I'm really touched by this. It is touching me also because it comes from this community. It's an incredible honor to receive an award, a medal from the people who work in your field, who are best to understand what it means, and sometimes even better than me, frequently better than me. So I would like to thank, of course, the committee, you, Harry, for the very, very, very kind words, but the committee who made the choice, the organizers of the CBM meeting, you know, and they must be commended also for organizing a CBM meeting during the pandemic. Must be nerve-wracking there. So thank you also, Morten, Vincent, and the Norwegian colleagues. Thank you to the committee. Beyond the 14th CBM meeting, I would like to thank Birte Svensson and our two colleagues, the Pedersens, Sven and Stefan, who helped her organize the first version of the CBM meeting there. And without the CBM meetings, I'm not sure this field would be what it is, regardless of everything there. So I'm happy to really thank Birte for her great contribution to the field there. I have to have a word from my employers. I would like to thank the National Research Center of France, CNRS, for the freedom I had throughout my career, and a freedom that was instrumental to my work. Freedom meaning not being interested at all. But, you know, freedom is freedom, you know, so you got to be appreciative of the luck you have there. So I was extremely lucky that CNRS let me do what I wanted and did not guide me on a dead end like they are very good at. I have to thank also, and no, more seriously, the French INRA organization, the INRA-E, as they call themselves now, because they've been particularly generous and helpful, and they have let one of their engineers work with me, Elodie Drula, work in my team in Marseille for over the years there. So thank you, INRA. Of course, I would like to thank my current employer, the Technical University of Denmark, who's giving me now the opportunity to continue my quest for the special relationship between sequences and functional features of K-zymes there. And of course, nothing would have been possible alone, you know, and I would like to thank, I'm so privileged, so lucky to have found all the wonderful people who have been and are still working so hard at maintaining the KZ database, despite the lack of direct funding and despite various initiatives that attempt to copy our assignments, but without participating to the hard day-to-day maintenance work there. So the team in Marseille is now in the hands of a wonderful young scientist, Nicolas Terrapon, who is more capable than me to meet the bioinformatic challenges of the next decade. So thank you, Nicolas, Elodie, Pedro, Vincent, Marilyn, for your incredible commitment and hard work. And thank you all for liking KZ.